Genetic profiling. Okay, so this is where it's used in forensic science to determine if someone's DNA, which is left at a crime scene, belongs to that person. Sometimes it's called genetic fingerprinting, sometimes called DNA profiling or DNA fingerprinting. It's all the same thing. Now you may think, well, everyone's DNA is different, so you could just sequence the whole thing, find out the base sequence of the whole thing, and compare it. Well, obviously, that wouldn't be. Well, it'd be possible now, but like even 10 years ago, it would not have been possible. And also, it's like a sledgehammer cracking a nut. There is easier ways of doing it than that. Now, the specification wants you to know about this method, which is called restriction fragment length polymorphism. Okay. Polymorphism. Now, polymorphism means that one gene can have a lot of different alleles in different people. So, hair color is polymorphic, red, brown, blonde hair, and so on. So this is to do with the the length of the bits of DNA which is produced. Well, first of all, let's just remind ourselves what a restriction enzyme is. A restriction enzyme, it's an enzyme used a lot as a tool in molecular biology. It's found in bacteria as a sort of defense mechanism, but it's a restriction enzyme that will cut DNA at certain sequences, okay? And those sequences that it recognizes are usually 6 to 8 bases long. So wherever it sees that particular sequence, it will bind to the DNA and chop it. Okay, so and so the restriction enzymes produce these fragments, and they have different lengths, and hence the polymorphism bit. Okay, so let's imagine we have 2 bits of DNA from, let's say, a bacteria, a fairly small bit of DNA. So this is from one bacteria, and this is from a very close relative of it, the same species, the same strain, but just a different bacteria. And let's call that X and Y. Okay, so let's say that X has got a restriction site there, so it's got the site that the restriction enzyme can bind to there, whereas Y has it here, slightly different place. Right, so if you cut those 2 things, let's say that this is, for example, let's say this is 6 kilobases long and this is 7 kilobases long. And let's say, for the sake of argument, that's 3, and so that would have to be 10, the same length, both 13 kilobases long. Okay, so if you chop those 2 bits of DNA from, so you had like a culture of bacteria X, a culture of bacteria Y, you extracted the enzyme, sorry, you extracted the DNA, which is pretty easy to do, added the restriction enzyme, you chop it up, right? X, you'd chop it into a 6 and a 7, and Y, you chop it into a bit 3 long and another bit 10 long. So let's say then, how can you find out the sizes of those? You could put it on an agarose gel, do electrophoresis. So here's our agarose gel. As we've said before, DNA is negatively charged, so you've got a positive voltage up here, okay, negative voltage there, and the fragments are all going to move that way. The smallest fragments move the fastest. So we put our X into there, put our Y into there. So X is going to give us, right, it's going to give us a fragment in different colors. One at 7, which doesn't go that far, and 6 goes a bit further because it's smaller. If you did that with Y, you'd find out you'd get this big fragment at 10, and then the 3 would go a lot further. So you get a different pattern of bands on your gel agarose gel electrophoresis. And so you're distinguishing by the 2 bits of DNA by the different lengths of the fragments produced. Okay, so if you did that with a human being, okay, so let's do, if you did that with a human, so you got some human DNA, extracted all the DNA from some cells, and you run that on an agarose gel, what you would get, and you treat it with a restriction enzyme, you'd get a smudge like that, because you produce so many different fragments and you wouldn't be able to see what on earth was going on. You get fragments of all those different lengths there. So imagine this is our bit of human DNA here. I'll do ... Okay, it's extremely long, much longer than a bacterial DNA, of course. Okay, and also it's in several bits, because it's there are 23 chromosomes, 23 pairs, so 46 chromosomes, but not worrying about that. So that's why you're going to get a huge amount of smears. But what you could do to reduce the number of bands that you're actually detecting is you could use a radioactive probe. Now, a radioactive probe, I'm going to draw it like that. And I'm showing that to show the sequence of the bases there. Maybe you've got 20 bases. And that can only H-bond to a specific sequence of DNA. It won't H-bond unless you've got a complementary sequence, which you have there on this bit of the DNA. It's not going to hydrogen bond. So you get your agarose gel, and you blot onto a piece of filter. So you blot the DNA, and that's called a southern blot. You may have heard of a southern blot. Just blot it onto the filter, and then you add your radioactive or your fluorescently labeled, this is called a probe. It's a little sequence of DNA which has been labeled with fluorescent things or with radioactive phosphorus. Okay, and you let that go there. Right, and then you'd see the pattern of radioactivity. It only binds to those fragments which contain that sequence. So you're going to reduce the number of fragments it identifies to a much smaller level. So I've drawn there 4. Now, if you could do that, then you might be getting somewhere. You might be able to distinguish between 2 different people's DNA from the size of the restriction fragments. Okay, now someone has been a lot cleverer than that. In 1985, Alex Jeffreys in the University of Leicester was the first person who developed this technique. And what he looked at, he realized, he knew there were certain sequences in human DNA. As you know, something like 98% of human DNA is non-coding. And a lot of that is kind of quite repetitive stuff. And there are things called variable number tandem repeats. And what that means is bits of DNA, you've got a normal bit of DNA in black there. And then this is, you've got this repeat bit. And this repeat, they can be between 6 to 100 bases long, these repeats are. So let's say we've got a little sequence there, which say, 10 or whatever, doesn't really matter. So that's repeated, okay? Right, so let's say in one person, you're going to get 4 repeats, okay, and 1 bit of DNA. Now what causes these repeats to arise actually is quite interesting, is because when the DNA is being copied, right, so there's your bit of DNA. Here's the new strand of DNA being made. The DNA polymerase starts going this way, and then it may detach. It may be detached from the template strand, and then what can happen? Then it can reattach, but reattach, and it forms hydrogen bonds with the wrong bit because it's just repeated all the time. So it might do that. Okay. So it's formed an extra loop, but it can still hydrogen bond here because you've got the same sequence there as you've got in the bit that's looped out. Okay, so it wouldn't realize it made a mistake, the DNA polymerase, if it did that, and that's what tends to happen. So Usually the case is that the number of repeats is getting bigger due to this looping out. Right, so that's why different people will have different number of repeats, and the number of repeats, generally speaking, doesn't make any difference to the function of the DNA. It doesn't seem to make that much difference. So it's not selected against in natural selection. So different people have different numbers of these repeats. If you got this kind of process, some kind of mutation occurring in hemoglobin, that wouldn't last. That the offspring produced would be non-viable and would die. And so you don't get mutations like that in genes which code for proteins. But in these non-coding regions with these repeating bits, it's very common. So everyone's a little bit different in that respect. Okay, so let's talk. Let's say we've got person A, and we've got this one particular tandem repeat. So whatever it is, we said it's 6 long. There's lots of different ones, right? So let's say this is person A. Now, don't forget that people are we're diploid. So on one chromosome, you're going to have the other chromosome. So person A might have that from the mother, and on the other one, let's say they've got so let's say they've got 4 there, and let's say here on this chromosome they've got 10 of them. 1, 2, 3, 4, 5, 6, 7. Okay, that's 10. And there is in the flanking sequence, so they're not in the, you don't want a restriction enzyme site in the repeats, but let's say we've got a restriction enzyme site there in green and a restriction enzyme site there. And in this chromosome, you've got it there. Let's say you've got another person, you've got person B, and he's got slightly different numbers. He's got, let's say he's got 9 and 16, okay? So let's go. So he's got 9 of those repeats, 1, 2, 3, 4, 5, 8, 9. They should all be joined together, but it takes too long to draw them. And then you've got 16 here. Well, I'm just going to guess 16. Okay, I'm not going to count them all. So he's got 16 in that chromosome. Okay, and here, got the restriction sites in the flanking sequence, the same as in individual A. That's more conserved. That bit stays the same. Okay, so now if you cut that with the restriction enzyme, you're going to see that this A, well, you're going to get a fragment, let's call it 4 units long, however many kilobases it is or whatever, 4 units long, and this one is going to be 10 units long. Whereas in person B, you're going to get one which is, what did I say, it was 9 and 16. It's going to be 9 units long and 16 units long. Now, if you put that on a gel and run it, you're going to have the same problem of all that. You know, you're going to have so many different fragments. You're going to get this problem. Here's your gel, where you're just going to get a, you know, a complete smear. So you're going to have to identify the fragments you're interested in by using a probe. Let's do the probe in yellow. Now the probe would probably be, well, it's a complementary sequence to the repeat, okay? And so it can, if you, when you, so here's your gel. Let's say that's person, let's call it person A, that's person B. So you run your gel and you get a smear. Of course, you then transfer this to a nitrocellulose. So the gel, then you transfer your DNA to nitrocellulose filter. To a filter piece of filter material. Okay, that's called a southern blot. And then you add the probe, which can only hydrogen bond to the bit in the repeat. So that binds the repeat. Okay, so we did person A and person B. Right, person A, you're going to get one fragment, which is going to be 4 units, which is going to move up quite a long way because it's small, moves fast. and you're going to get a bigger one at 10 there. Person B, you're going to get one at 9 units. That's going to be a bit further than the 10. You get a band there, and then you're going to get the 16, which doesn't move so far. So you've got a different pattern of bands on individual A and individual B. And also, interestingly, you get 2 bands for each person because we are diploid. Now, that is not going to be enough to identify someone unambiguously. You know, if you found someone's DNA at a crime scene and it was like that, and then you tested somebody who was a suspect and they had the same, well, there is a coincidence, there is a chance that it just could be pure coincidence. Let's say the chance of them being the same, let's say it's 1 in 100. So you choose 2 people at random and they've got the same, they'd give you the same restriction fragment of polymorphisms using that particular probe. All right, it's not very good, but you can make it much better. Like that's with 1 VNTR probe. So you're only looking at one. You're looking at 1 gene there. 1 gene locus, they usually call the locus is a place on a chromosome where the gene is. So what in Britain, they don't use 1, they use 13 different VNTR probes, which will go to 13 different loci. Now that then makes the probability much, much smaller. It will be, the probability would be 1/100th times 100th times 100, so on, 13 times. So it would be 1/100 to the power of 13, which is a pretty small number. And I've just made that 1 in 100 up. It may be the actual likelihood of being the same could be way less than 100. It probably is. So by the time you've used 13 different probes, the chances of someone having the same pattern of bands are going to be vanishingly small, impossible. So that is how our restriction fragment length polymorphism using variable number tandem repeats, DNA profiling works. And as we'll see in a later video, this is one example I'd want you to use about, but it's not actually it's not the method that's used mostly. There is a better way of doing it using PCR, polymerase chain reaction. It's a very similar idea, and people have different lengths of these repeats, but you identify the lengths of the repeats using the PCR process, which of course can use much, much smaller amounts of DNA. The problem with this original method was you needed a reasonable amount of stuff to actually do the experiment on, whereas with PCR method you can use vanishingly small amounts.